All these examples are taken from the NCERT textbook itself. Those only you have to use. You can't write some other example. So that is why they have made a textbook. You have to follow whatever is there in the textbook only. 2 times in meiosis 1 and meiosis 2 also DNA replication will not take place. Only once DNA replication will take place. Both of them bear seeds, that is gymnosperms and angiosperms. But why are they classified separately? For that you have to answer. If you write complex 1 and complex 2, complex 3, complex 4, maybe they might give you half a mark, but you will not get full mark because you have to name them. Hello everyone, a warm welcome to the session on discussion regarding the model question paper for first PUC Biology. I'm Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So this is the second half, that is the second session of the model question paper. So in the previous session, we had discussed about the blueprint and how the model question paper looks, and also we had discussed part A and part B that is given in the model question paper. We had solved the question and the answers. So it's a continuation in this session wherein we'll concentrate on part C and part D. So part C, it carries 3 marks. That is, it is a 3-mark questions will be asked in part C. And in part D, 5 marks question will be asked. Again, part D is split into section 1 and section 2. So all those questions we shall solve today. So here, answer any 5 of the following questions in 40 to 80 words each wherever applicable. So it is a 3-mark question. So 5 questions will be, that is somewhere around 8 questions will be asked, and out of the 8, you have to answer any 5 whichever you know perfectly. So the 29 questions, so the remaining questions were in part A and part C. So here the 29 question under part C, how ascomycetes are different from basidiomycetes. So ascomycetes and basidiomycetes, we had studied them in fungi, right? That is, fungi are divided into different classes: Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes, right? So here they have given, write the difference between Ascomycetes or Basidiomycetes, or I'll read the question as it is. How Ascomycetes are different from Basidiomycetes? The name itself says Ascomycetes. So whenever this word mycelium or mycetes, it comes, no, you have to understand it is fungi, okay? Because mycota means fungi. So therefore mycetes meaning fungi. Now asco and basidio. So it produces what? Ascospores. Basidiomycetes produces what? Basidiospores. And where are the spores produced? In the ascospores are produced in fruiting bodies called ascocarp. Basidiomycetes are produced in fruiting bodies called basidiocarp. So we shall look at it. So the fruiting body is a aci, or it is a ascus because it is cup-like in structure. The fruiting body is basidia. So whenever you have to write the differences, always make sure that you draw 2 columns. Don't write it in the form of paragraph. That is not the way of writing differences or differentiate between all that. You have, when it is asked, you have to make 2 columns like this. You have to, in one column, name Ascomycetes, another column name Basidiomycetes, and then go on answering. Again, when you're answering, let it be in the form of point. So when you're writing the first point, the next point that you write in the next column should be opposite of what you write in the first column. So that is how differences needs to be written, because the question itself is differences, right? You can't write whatever points however you want, you can't write. Say for example, here under Ascomycetes, the fruiting body is ascus. There it is, you have to write about the fruiting body only. You can't write about the spores there. Because why? Here you're discussing about the fruiting body, so you have to write opposite of that there. So that is why. So fruiting body is ascus, there the fruiting body is basidium. Here ascus produces ascospores, basidium produces basidiospores. This is one of the major difference. Here asexual spores are formed, meaning conidia are formed. Here asexual spores are not formed. So these 3 differences, wherein each of these will carry half a mark. So therefore here it is 1.5, and here half a mark each, 1.5, total 3. So 3 points on either side you have to write if you need to get 3 marks. Next question. Coelenterate showed two basic body forms. Explain with an example each. So coelenterates, intercoelenterates, sometimes in the paper they can ask as cnidaria also, because coelenterates are nothing but cnidarians. So they show two basic body forms. What are those? You need to explain. So the two basic body forms that they show is polyp, and then one more is medusa. So write it as A and B, polyp, medusa. Let it be very neat, and so that the examiner, it is easy for him or her to correct as well. Polyp, it is sessile and it is cylindrical form. Sessile means they are not capable of moving. They are stagnant. They are sessile and they are cylindrical in form. Example, what they have given, explain with an example. Usually what students do is they'll just explain and they'll forget to write the example. No, they have mentioned in the question there, explain with an example. So one example should be there in each of the forms here. Next, medusa, they are free swimming and umbrella shaped. Example, Aurelia, or it is also called as jellyfish. So therefore, you write this. It will carry 1 mark, and one example you write, it will carry half mark. Therefore, 1, 2, 3. 3 marks. This is how the marks distribution is there for this. Moving further. Classify the flowers based on the position of ovary. Based on what? Based on position of ovary. You can classify the flowers based on the estivation, or based on the number of the attachment of the stamens, like that, and all classification is there. So read the question properly. Here what have they asked? Classify the flowers based on the position of the ovary with respect to other floral whorls, mentioning one example. Here also mentioning one example each you need to write. So here usually the flowers are based on the position of the ovary. The flowers are classified into 3 types: hypogynous, perigynous and epigynous. Hypo means always on top of something, above. So therefore hypogynous means ovary superior, wherein all the other floral whorls that are there, they'll come from the base of the ovary. So therefore ovary is in the superior position. Example, you can write any one. 3 I have written here: mustard, China rose, brinjal. Out of these, any one you can write. Next one, perigynous. Here ovary is in the center and other parts of the flower or other whorls of the flower, they come from the center of the ovary. So therefore the ovary is partially superior or partially inferior. Therefore, example here for this is plum, rose and peach. These are the example. Any one example you can give. So how neat it looks. No, if you write A, B, C, write the side heading, then explain it, write an example under it. It looks so neat. So this is how you have to answer the question. Next, the third one is epigynous. Here ovary is completely inferior. Therefore, all the floral whorls that are there or the floral parts that are there, they come from the top of the ovary. Therefore, the ovary being completely buried. So it is inferior ovary. Example, guava, cucumber, ray flowers of sun or ray florets of sunflower. All these examples are taken from the NCERT textbook itself. Those only you have to use. You can't write some other example. So that is why they have made a textbook. You have to follow whatever is there in the textbook only. Moving further. Write a note on the vascular bundles of root and stem. Here you have to write a note on the vascular bundles of both the root and the stem. So monocot root, you have to write about the monocot root because they have not mentioned there anything. They have just said root and stem. But in your NCERT textbook, they have described about the vascular bundles of monocot root, dicot root, monocot stem and dicot stem. So you'll have to write about all those. Dicot root, then monocot stem and dicot stem. We'll see one by one. So monocot root, more than 6 xylem bundles. That is, they exhibit poly-R condition. That is one of the peculiar characteristic, right? Whereas in dicot root, only 2 to 4 xylem and phloem patches are present and they are conjoint open type vascular bundle. Next, talking about monocot stem, here also vascular bundles are conjoint and closed. And the peripheral this is one of the peculiar characteristics wherein the vascular bundles which are situated at the periphery, they are smaller than the ones at the center. So that is also one of the peculiar. Now in dicot stem, vascular bundles are arranged in a ring-like manner that is in the form of a ring, and they are conjoint open type. So if you write one point under this also, it is enough. So this is about the vascular bundles of root and stem. So very easy. You don't have to write half a page or anything because it's a biology subject. You have to neatly write with side headings, subheadings, and the points under it. Next question. Name the enzyme involved. See here, they have asked 3 questions here. Each question will carry 1 mark. 3 questions they have asked. Name the enzyme involved in breaking of carbon-carbon or phosphorus-nitrogen bonds. And next question is, name the enzyme involved in hydrolysis of substrates by leaving double and triple bonds. Name the enzymes involved in catalyzing the joining of carbon-oxygen and carbon-sulfur bonds. So here for each one, if you just write these answers, you will get totally 3 marks because all the 3 you have to answer. So the enzyme involved in the breaking of, you don't have to write the enzyme involved in the breaking of carbon-carbon or phosphorus-nitrogen bonds are hydrolases. No, just write A, write hydrolases, write B. Before that you have to write the question number 33, then write A, write the answer, write B, the answer, C, just the answer in a word. It is enough. For A, breaking of carbon-carbon or phosphorus and phosphate-nitrogen bonds are done by which enzyme? Hydrolases. The hydrolysis of substrates by leaving double and triple bond is done by which enzyme? Lyases. And ligases is involved in the catalysis, or it is involved in the joining of carbon-oxygen and carbon-sulfur bonds. So you write each of the name the enzyme, you will get 3 marks. Next question. Here also it is split. 1 mark, 1 mark, 1 mark. So all the 3 you need to answer. So this is an analysis kind of question. So meiosis involves 2 sequential cycles of nuclear and cell division. That is true, right? Because in meiosis we have meiosis 1 and meiosis 2 are involved. So therefore, how many sequential cycles are there? 2 sequential cycles. Called meiosis 1, it's here, meiosis 1 and meiosis 2. But only a single cycle of DNA replication will take place. 2 times in meiosis 1 and meiosis 2, also DNA replication will not take place. Only once DNA replication will take place. With reference to this, in which sequential cycle of meiosis all these 3 processes will occur, you have to write. So first one, synopsis and recombination. That is in meiosis 1 and meiosis 2, or in meiosis, in which stage of the cell division does synopsis and recombination occurs. Okay. So here synopsis is nothing but the synapto-nematal complex that is there, that will get formed, which is a bridge-like structure that joins the chromosomes. And recombination, that is one chromosomal arm will recombine with the other. So where does it occur? It occurs in the zygotine stage. Next, 2n number. 2n means diploid. So diploid number of chromosomes becomes haploid number. It becomes haploid number. In which stage does this happen? It occurs in telophase 2. So in telophase 2, what happens? The chromosomes which were diploid, they will become haploid. Next, 4C DNA becomes 1C. So this occurs in the diplotein stage, wherein 4C DNA becomes 1C. So this is these 3 you mentioned, you will get 3 marks. Moving further, what is oxygen dissociation curve? So here, if you write what is oxygen dissociation curve answer for this, you will get 1 mark. Which factors result in the dissociation of oxyhemoglobin? So 2 factors if you write, you will get 2 marks. So therefore total of 3 marks. So this is how you can answer. First you need to write what is oxygen dissociation curve. Oxygen dissociation curve, or it is a curve or it is a graph representing the percentage of saturation of hemoglobin with oxygen, which is plotted against partial oxidation. So which is plotted against partial oxidation of oxygen. So PO2 means partial oxidation of oxygen. So that this much you write, you'll get 1 mark for this. You write the definition. Now you have to write 2 factors resulting in oxygen dissociation curve. So first factor is high partial pressure of oxygen, low partial pressure of carbon dioxide, lesser H plus ion concentration and lower temperature in the alveoli should occur. Okay. And next, for the low partial pressure of oxygen, high partial pressure of carbon dioxide, high hydrogen ion concentration and high temperature in tissues should occur for the dissociation. So 2 factors, formation and dissociation should occur. So formation, you have to write these are the factors. For dissociation, these are the factors that are involved. So 2 points you write, you will get 1 mark for each point. Therefore, totally 3 marks. So this is how you have to answer. So moving further, draw a labeled diagram. Here itself it is given. If not here, don't think that if they just ask draw a diagram also, you have to label, because first itself in the instruction you have seen in the previous session, right? In the instruction they would have shown diagrams without label will not fetch you any marks. So labeling is very important. Draw a labeled diagram of axon terminal and synapse and label the following. So we have to label all of this. So they have given already what labeling should be there. So presynaptic membrane should be there. So diagrams should be taken from the NCERT textbook itself. Diagrams other than that will not be entertained. What is there in the NCERT textbook? Same diagram you have to draw. So this is the synapse that is given, axon terminal and synapse that is given in the NCERT textbook. The same thing you have to draw. So what all should we mark here? Presynaptic membrane should be there. So presynaptic membrane, this you have to mark. Then synaptic cleft, it has to be marked. And then postsynaptic membrane should be marked. All this should be there in the diagram also. And receptors should be marked. These are the ones which receive the neurotransmitters. Next, neurotransmitters should be marked. They are the chemicals. Next, synaptic cleft already. Okay, neurotransmitters, then axon terminal. So this one. So much you have to mark. So say for example, it will carry, if at all it carries half mark here, half each. So 1, 2, 2.5 plus half for the diagram. Maybe that is how it can be divided, or according to the scheme, quarter mark also can be given for labeling. So it depends on how the scheme is being set. So this is how all the labeling should be done. Or else, if they don't give the names of the label, still, because it is a 3-mark question, 6 labeling should be there. That is how it is. Next question, that is in part D. So we'll discuss what and all questions are there under section 1. Here, answer any 4 of the following questions in about 200 to 250 words each, wherever applicable. Any 4 if you answer, it is enough. Again, here it is divided as, so 2 questions have been asked under this particular question number 37. That is one question here and the other one is B. It is here. So first question, that is A under 37. What is heterospory? And you have to comment on its significance. So what is heterospory? Heterospory is a condition in which the pteridophytes, so spores are produced in pteridophytes. So pteridophytes produces 2 kinds of spores which are different in their size. One is macrospore and the other one is microspore. That is called as heterospory. So next question, both gymnosperms and angiosperms bear seeds. Then why are they classified separately? So both of them bear seeds, that is gymnosperms and angiosperms. But why are they classified separately? For that you have to answer. So gymnosperms and angiosperms bear seeds but are classified separately because gymnosperms have naked ovules, that is their seeds are not covered, whereas angiosperms have seeds which are enclosed in fruits. That is why. And in gymnosperms, the pollen grains and the ovules are developed in micro and megasporangia like structures, or cone-like structures, but in the case of angiosperms, they are developed in flowers. So this also is one of the important. So much you write, you will get 5 marks. Next question. Compare the characters of non-chordates and chordates. This is one of the common questions that keep repeating every year. So as it is in the textbook, if you write, it is enough. So chordates, notochord is present. So here 1, 2, 3, 4, 5 points you need to write on each side to get 5 marks. Here notochord present, here notochord absent. Under chordate, central nervous system is dorsal, hollow, and they have a single central nervous system. In non-chordate, central nervous system is ventral, solid, and they have a double central nervous system. Next, chordates, pharynx perforated by gill slits, but in the case of non-chordates, the gill slits are absent. The pharynx are not perforated. Next, in chordates, the heart is situated in the ventral. In non-chordates, the heart is situated in the dorsal side. Next, in chordates, post-anal tail is present, but in the case of non-chordates, post-anal tail is absent. So much you have to write. So next question, draw a neat label diagram of, here 2 diagrams are given. In a hurry, just don't draw one, chloroplast and cilia, and label the following that are related to chloroplast and cilia. Now you have to pick which label comes under chloroplast and cilia, and then you have to segregate and label it properly. So this is also the diagram that is taken from the NCERT textbook. So whatever labeling is there, that needs to be labeled. So this is how you have to write this. So next question. Here again, 2 questions are asked under question number 40, question A and question B. Name the 5 complexes that are involved in electron transport system of inner mitochondrial membrane. You just name the complex, it is enough. One is NADH. You can't write it as complex 1, complex 2, complex 3, complex 4, complex 5. No, you have to name the complexes. Here, first one, NADH dehydrogenase complex. The second one is cytochrome BC1 complex. Third one is succinate dehydrogenase complex. Fourth one is cytochrome C oxidase complex. Fifth one is ATP synthase complex. So you have to name the complexes. If you write complex 1 and complex 2, complex 3, complex 4, maybe they might give you half a mark, but you will not get full mark because you have to name them. What is the next question here? So you have named them. Here next question is mention the cytochromes that are present in the complex 3 and complex 4. Say for example, each of these might carry half mark each, half. So totally how many? 1, 2, 2 and a half. So for this 2 and a half, next what is the next question here? Mention the cytochromes that are present in the complex 3 and 4. You have to mention the cytochromes that are there. And the number of protons that are pumped by these cytochrome complexes is the one that you need to mention. So complex 3 and complex 4 is what they asked, right? So complex 3, the cytochromes that are present in that is cytochrome C1 and cytochrome B. In complex 4, it is cytochrome A and cytochrome A3. And number of protons, so here we have half. And number of protons pumped through these complexes is complex 3 will pump 4H plus protons, complex 4 will pump 2H plus proton. So you write this, you will get half a mark. So therefore here again, the mark division depends on the scheme, how the scheme is set. Next, moving further, the role of abscisic acid and ethylene is both positive and negative. Justify the statement. So you have to write the positive and negative aspects of both abscisic acid and ethylene. So first what we'll do is, we'll write the positive role of abscisic acid, then we'll write about the negative role of abscisic acid, then move on to writing the positive and negative role of ethylene. So positive role of abscisic acid, when a plant is stressed, the concentration of abscisic acid in its leaves will rise, causing the stomata to close, reducing water loss by transpiration. So this is one of the positive aspects. Negative role of abscisic acid is it inhibits the growth of plant and therefore it causes seed dormancy or dormancy in that particular plant. Next, we'll move on to ethylene. Positive role of ethylene. Ripening of fruits, it stimulates the creation of root meristems and the growth of lateral roots when present in lower concentration. Negative role of ethylene, it is responsible for nullifying geotropism. So much you have to write. If you write the positive and the negative aspects of both these hormones or growth regulators, you will get 5 marks. So next question. Draw a neat label diagram of functional unit of kidney. See here, they have not asked nephron directly. They have asked functional unit of kidney. So be very, very careful. So which is the functional unit of kidney? It is nephron. So therefore, you have to draw the structure of the nephron and label. At least 10 labeling should be there because it is a 5-mark question. So draw it very neatly. Next question. Differentiate, they have asked 2 questions, question A and question B under 43rd question. Differentiate between A band and I band present in the myofibril. And one more, name the joints present between, again they have divided here. So this will carry 2 mark, this one 1 mark each, therefore 5 mark. So how they have split, you can see here. Name the joints present between humerus and pectoral girdle, carpal and metacarpal of thumb, atlas and axis. So first we'll answer the first question. Again it is write the differences now. So you have to make 2 columns, A band and I band. A band, it is the light band in the muscle fiber, and I band is the dark band in the muscle fiber. A band, it is made up of actin proteins and hence it is also called as actin filaments. Here I band is made up of myosin proteins and hence it is also called as myosin filaments. And A band is an isotropic band, whereas I band is an isotropic band. So much if you write, that is here you will get 2 marks. So you can write just 2 difference under each of these, it's fine. So 2 marks you will get. Next, so you have to write, name the joints present between humerus and pectoral girdle. The joint present between humerus and pectoral girdle is called as ball and socket joint. The joint present between carpal and metacarpal of thumb, it is called as saddle joint. And the joint present between atlas and axis, it is called as pivot joint. So you write each of these 1 mark, you will get totally 5 marks. So next question. Here again, 2 questions have been asked under question number 44, question A and question B. Mention the 3 layers of adrenal cortex and the corticoids secreted by them. You have to write the 3 layers and you have to write about the corticoids that are secreted. So inner layer, zona reticularis, middle layer, zona fasciculata, and outer layer, zona glomerulosa. And you have to write about the corticoid that is secreted. Next question. Thyrocalcitonin and parathyrohormone plays a significant role in calcium balance in the body. Comment on that. So thyrocalcitonin secreted by the thyroid glands, they actually regulate the blood calcium levels and they also regulate the secretion of parathyroid hormone by the parathyroid gland, and this is regulated by the levels of calcium ions in the blood, and this calcium ions in the blood in turn, it will increase the calcium ion levels in the blood and thereby its absorption by the bone. So that is its function. So you write so much, you get 5 marks. So next we'll move on to section 2. Answer any one of the following questions. So 3 questions will be given, any one you need to answer. So each of 45th question, each of membranous organelles is distinct in term of its structure and function. Many of these are considered together as endomembrane system, right? So few of the organelles have been put together under the endomembrane system because of their functions, because the functions are coordinated. Now you have to give reasons, you have to substantiate the statement. So while each of the membranous organelles is distinct in terms of its structure and function, still many of these organelles are considered together and been put under an endomembrane system as they share a structural continuity. What is the structural continuity? Let me see. That is endoplasmic reticulum and Golgi. So we have endoplasmic reticulum, then the Golgi bodies, then the ribosomes, lysosomes, all these are put under the endomembrane system itself. Why is it? So here endoplasmic reticulum and Golgi apparatus, they play very important roles in packaging, modification and transport of proteins and lipids within or outside the cell, because we know that endoplasmic reticulum will give the proteins and all that to the Golgi apparatus, which will modify the proteins into glycoproteins and lipids into glycolipids. So therefore both are interconnected to one another. Next statement, that is endoplasmic reticulum, especially the rough endoplasmic reticulum, is the one that synthesizes proteins, and whatever protein it synthesizes, it is modified and folded within the endoplasmic reticulum before it is being transported to the Golgi apparatus, wherein in the Golgi apparatus these proteins get sorted. So therefore both are interconnected to one another. And lipids are modified in smooth endoplasmic reticulum. Then lysosomes, what is there? It will break down waste and cellular components, and it also helps in the coordination of cellular functioning. So that is why all of these lysosomes, ribosomes, Golgi apparatus, even the vesicles also, and as well as the endoplasmic reticulum, they are placed under the endomembrane system for this particular reason, because each of them depend on one another to perform a particular function. Next, 46th question. What special anatomical features are displayed by leaves of C4 plants, and how do they provide advantage over the structure of C3 plants? So in C4 plants and C3 plants, we know that the process of respiration is different, right? How is it? So in C3 plants and C4 plants, we know that the process of photosynthesis is different, right? So why is it so? Because of some structural modifications. What are those? Let's look into. So the anatomical features displayed by the leaves of, there is no need for us to write about the differences in the structure of it. We have to write about the differences in the anatomical features. Say for example, they are capable of tolerating high temperature, and they show response to high light intensities. Next, they lack photorespiration and have greater productivity and biomass. Because they lack photorespiration too much, they have greater productivity of biomass. That is, their dry weight is quite heavy. And advantage, and we have to write about the advantage of C4 plants over C3 plants. So one of the main advantages, Kranz anatomy, that is, they have a sheath of large cells which surround their vascular bundles. So in the leaves of C4 plants, if you have a look, their vascular bundles are surrounded by few layers of cells, and they're called as the bundle sheath, and it is nothing but the Kranz anatomy, and this helps in increasing carbon dioxide concentration in the leaves. And therefore relative concentration, because carbon dioxide is one of the major factors which are responsible for the formation of glucose during photosynthesis. Next, relative concentration of carbon dioxide and oxygen affects binding of a particular enzyme called as rubisco to a particular molecule, and high carbon dioxide in the leaves of C4 plants, it ensures that all the rubisco binds with carbon dioxide and not with oxygen. Thus, photorespiration does not take place, and therefore the biomass is more of this particular C4 plant. So therefore, C4 plants have more advantage over the C3 plants in terms of photosynthesis. So this is much you have to write for this particular question. Next one. The sequential events in the heart, which is cyclical, repeated, is called the cardiac cycle. Explain these sequential events that in turn is responsible for double circulation. So you have to, it's nothing but they'll just give such a big question and make you confused. Only thing is here you have to write the events that occur during the process of double circulation of the heart. That is what you need to write. So the cardiac cycle occurs in two phases. One is you have to write about the contraction, that is systole, and you have to write about the relaxation, which is the diastole. So we'll write about what happens when there is relaxation in the auricles and the ventricle, and when there is contraction in the auricle and the ventricle. So first we'll start talk about the atrial systole. Atria means nothing but it's auricle only. So So during the contraction of the atrium, what happens is both atria contracts and it forces the blood from the atria into the ventricles. Now the blood has gone to the ventricle. Now the ventricles will contract. What will happen in the ventricle? We will look into. That is ventricular systole. Here both the ventricles contract, and when it contracts, the blood is forced to the lungs via the pulmonary vein and then to rest of the body through the aorta, which is the major vein. Next, what happens during relaxation? That is in the atrium and in the ventricle. So atrial diastole. Here, relaxation of atria, during which atria fills with blood from the vena cava. That is from the superior and the inferior vena cava. Next, in ventricular diastole, that is when the ventricles are relaxing, what happens? It actually begins before the contraction of the atria or the auricle, thereby allowing ventricles to fill with blood from the atria. Next, we have to move on to double circulation. So the right ventricle that is there, it will send deoxygenated blood to the lungs, where the blood becomes oxygenated, and that oxygenated blood will return back to the heart. And left ventricle sends newly oxygenated blood throughout the body, but the time this blood returns to the heart, it has returned a deoxygenate. That is, it will return in a deoxygenated state. Why it goes in a deoxygenated state? Again, it has to get filled back with oxygen. So much you have to write. That is, you have to write about the contraction and relaxation. However you want, you can write. But to make you easily understand, I have written contraction separately, relaxation separately, and then went in for double circulation. So that is how I have split and wrote the answer here. So this is how you have to solve the question. So this was about the session. So I hope the first session as well as this session was quite useful for you, because you got to know how you need to answer the model question paper. And it's quite important because some of the questions from this are can appear for the exam also. It may not, or it may. So this will help you and it will guide you how you need to answer the questions in your final exam. So I hope you found the session useful. Thank you.